we're humans on this earth in this universe, and it's great to try and understand more about it, even if we're not solving a problem. And I think basic science is valuable for those two reasons, because it can be a stepping stone to larger discoveries, but also because science for its own sake is valuable just for our own, I guess, like sense of wonder and like questioning about the world around us.
Hello and welcome to the STEM and Research podcast. Today we have Claire with us, here to share her story with science.
Claire is interested in the biological sciences and is a student researcher from Virginia. She is an ISEF 2020 finalist and a two-time winner at the Virginia State Science and Engineering Fair. Hi Claire, thanks for being here.
Hi, thank you so much for having me.
So I guess let's get started with this episode. First, we're just going to talk about how you got into research. So maybe you could talk about the starting point of what got you interested and how did you go ahead with it?
Yeah, so my school has like a course, like a research course where students are encouraged to do an independent research project. So I live in Blacksburg, Virginia, which means that Virginia Tech is really close by. So I had known about this opportunity and I decided to try and get a spot in a lab at Virginia Tech before my junior year so that I could do a project during my junior year. So the summer before my junior year, I just sort of cold emailed a few professors that I'd looked into and thought that their research was really interesting. And they all said no, which was great. But they had good reasons for saying no. Like they didn't have enough space in their lab, or they couldn't take on a high school student who would need a lot of mentoring, which was totally fine. But one lab said no, but you can join our journal club, which is like a weekly meeting where we discuss scientific papers. So I said, okay, any science experience is fine, even if I can't do research, this should still be pretty fun. So I joined their journal club, and after the end of the first discussion, one of the undergrad students said that he would take me on as a mentor. So that's sort of the weird, that's sort of the weird story of how I got into my lab. But it was a great experience. So junior year, I was able to do a project under that student. And then I stayed in the same lab to my senior year, then graduated, and now I'm working virtually for that lab over the summer.
Mm-hmm.
So my story of getting into science is, I guess, sort of perseverance and luck in combination, which I think is true for a lot of people. But.
Mm-hmm. So to me, it sounds like a gradual process, but from your personal viewpoint, would you say there was a defining moment, or was it more of a gradual thing?
For my interest in research?
Yes, the interest part.
Yeah. I mean, I would say it was definitely gradual. I came into high school thinking that I wanted to be an art teacher, and
Okay.
and then I sort of got more and more interested in science to the point where I thought I'm a high school student, but I'd love to have the chance to make a really cool discovery and see what a life in science would be like. And I thought that research was the best way to do that. So it was definitely sort of a gradual building of interest. I think I took AP biology my sophomore year, and I had a great teacher. And that really sort of kindled my passion for biology. So I decided wow, I'd love to work in a real biology lab and do the techniques that I read about in my textbooks. And then, yeah, all the professors that I reached out to had labs associated with biology or cell biology. And then I got in. So gradual building of interest, but definitely a very sudden and defined entry point.
Okay. So what are the fields you're most passionate about? You mentioned you took AP biology and most of your work has been in the biological sciences. Are you passionate about any other fields, or is this the one that you want to enter?
Yeah, so I've done all of my research has been cell biology and fission yeast specifically. So looking at mechanisms in fission yeast. So I'm definitely like really passionate about biology, but I'm not 100% sure what I want to do. I really liked this summer I worked virtually, and I really liked the process of like data analysis and, you know, getting a big data set and then looking through it. It's like a treasure hunt to try and find useful information or like answer questions. So I'd love to explore that more in college. And I really like the idea that data, like there are so many different types of data, and if you know how to analyze it, you can come up with solutions to so many problems and learn more about so many different topics. So that's sort of where I'm leaning right now, but I also really love biology. I kind of love all of science and engineering. I thought for a while that I was going to go into college and do engineering because I liked the idea of like really solving problems. I did basic science research in high school, which was a great experience, and like I love, you know, doing science for science's sake. I think that's a beautiful thing. But I sort of wanted to work in a field where I was like building tangible objects or like really making a tangible impact. But now I'm leaning again more towards the like data computational fields. So I've bounced around a lot and I'm still not really sure, but yeah.
Okay, let me pick one of the phases that you went through. Why do you think science for its own sake is a beautiful thing? Can you speak more about that?
Yeah, so I think like basic science is super important. A lot of people say, you know, basic science is important because without basic science, we wouldn't have the big discoveries because we have to learn about a system or a problem through basic science in order to do the science that solves the problem or does something to the system. And that's 100% true. But I think it's also really important to remember like we're humans on this earth in this universe, and it's great to try and understand more about it, even if we're not solving a problem. And I think basic science is valuable for those two reasons, you know, because it can be a stepping stone to larger discoveries, but also because science for its own sake is valuable just for our own, I guess, like sense of wonder and like questioning about the world around us. So yeah. And I think science fairs, and like in for research that students do, I think science fairs often sort of make kids feel like basic science isn't good enough because they're focused a lot on like impacts and applications, which is fine, because that's what science fairs like. You know, they like a headline of, you know, student develops, you know, a test or something, or like discovers a star, or you know, something like that, where it's like has a big impact. And you know, science with a big impact is awesome. But I think sometimes science fairs forget that like basic science is awesome too. So you know, when I was presenting, I felt a lot of pressure to, you know, go back all the time to like talking about diseases and things, even though, you know, I hadn't cured a disease. But you know, how my research could one day do this. But I think that's a pressure that we shouldn't have to put on our students in science fairs and sort of in research, because I think it's sort of unreasonable and it quashes the student's desire to discover just to discover, which I think is important.
Yeah, that's very well said. Anvesha and I was just discussing, yeah, we were just discussing about how, yeah, there's a lot of pressure in science fairs about uh, how you solve a particular problem more than what you did, and that, yeah, that definitely does create a lot of pressure. You're correct about that.
And I think
Yeah.
it's
So
really interesting to see how universal this is. I mean, even at the India Fair, we exactly faced the same thing. So we've pondered upon this for a while. So that was really well put.
yeah, and while you present it, even if two people have done the exact same thing and one of them chooses to focus on the impact that they have, they're going to get ahead more easily. That's one thing we've all noticed. Yeah.
Yeah.
So, what do you hope to accomplish with a career in STEM?
I mean, I think my main goal in my career is to do something that helps people, and I know that that's like a really broad thing, but that's sort of what I've wanted to do for a while. So like some people become, you know, doctors to accomplish this goal, um, or you know, or they do math research that will one day develop an algorithm that will save someone's life. But I I think for me that means looking at the big issues that our society faces and thinking about how I can address those. So in high school, I mean, I was looking at single cells, but I think in college, I'm really looking forward to studying not just science, but like the sort of social and societal sides of it to try and connect those two things and think about how I can use science to address these issues. But yeah, as I said, I haven't really done research on that scale to solve those sorts of problems yet, but it's something that I would want to do as a career.
Okay, and you're heading to Stanford, right?
I am, yes. Well, I'm not. Stanford is virtual
Yeah.
fall quarter, but yes, I will at some point set foot on Stanford's campus,
Yeah.
and I'm really excited to go there because I think they put a lot of emphasis on like their students not having to know what they do as soon as they come into college, which is great because I think a lot of other universities sort of expect students to know what they're going to do. And I mean, I know that I'm going to be somewhere in there, probably engineering or like a science related major in the College of Humanities, but I'm just not 100% sure. And I feel like very supported by them in that unsureness, which has been nice.
Okay, so let's talk about your work with the STE13 protein and its impact on certain mRNA, that is messenger ribonucleic acids. So can you describe this research?
Yeah, so like we all know, cells have DNA, and then they in order to turn this DNA into proteins, they have to make messenger RNA, which is like the precursor to proteins, goes out into the cytoplasm. Ribosomes get on this messenger RNA and translate it into proteins. So cells have to like really control the concentration of each protein that they're making because a lot of the processes that cells do depend on like how much of a protein is available. Like, if there's a lot of protein, it'll bind to a certain receptor and that will trigger something. But if they don't have enough of this protein, then that process won't happen. So one way for cells to like control how much protein they've got is to control how much messenger RNA they've got. And one way for them to do this is by having some sort of protein that binds on to certain messenger RNA and causes them to degrade. So there's less of this messenger RNA in the cell and then potentially less of the protein that makes. So I researched this one protein called STE13. And the reason why I looked at it is because in budding yeast, people had already found that its homolog, which is like a protein with a similar sequence, which people think will have a similar function, was doing that. So it was binding to messenger RNA, certain kinds, and degrading it. And so I thought, if this mechanism is conserved in fission yeast, budding yeast and fission yeast are both called yeast, but they diverged like 400 million years ago. So conservation in them indicates like conservation across all eukaryotes. So I researched its homolog STE13. And I found that if you take STE13 out of cells, then the concentration of messenger RNA is found to increase, which indicates that with STE13 in cells, it's performing a similar thing and causing these mRNA concentrations to decrease. So my research was sort of affirming that this protein is doing the same thing in fission yeast as it is in budding yeast. And then that it's probably doing the same thing in all eukaryotes. So we've got a version of this protein in us, which is probably doing that right now, which is cool to think about. But yeah, so that was my research about STE13, and it was sort of my first introduction to doing research. I will never forget when I saw the first graph that came out of the data and I was just looking at it. I don't think it was even like that meaningful of a graph. I think it was like just sort of to check that things were doing what they should. But I looked at this graph and I thought no one else in the world has seen this graph. And this graph is because of stuff that I did. And it was really meaningful and awesome.
Yeah, the small things definitely mean a lot along the way.
Yeah.
So why did you personally do this project?
So I like I said, I got into my lab under an undergraduate student, which I think is pretty typical. Like lots of students will have a mentor in the lab, especially because mine was a cell biology lab, so they have a lot of like equipment and imaging stuff and procedures that like I'd never heard of and needed to learn about. So I had a mentor that sort of guided me through the process, and he was looking at lots of things to do with messenger RNA and what's called codon optimality. So my research with this protein is that it binds to messenger RNA with lots of what are called non-optimal codons. So my research was sort of part of his work, which was great because I had like a mentor who, you know, knew what I was doing and was able to help me understand everything, you know, help me use the fancy microscopes and things. So it was that was sort of how I got into it and how I got into the lab.
So,
makes sense.
mhm. So I know you already mentioned the results, but maybe we could just specifically dive into it a bit. So, what did you find through this research? You could probably address this on a personal level as well if you wish to do so.
Yeah, I mean, so like I said, found that without ST13, there's more of these messenger RNA, which indicates that with ST13, there's less. So ST13's function is probably to lessen the concentration of these mRNA. And so that was like my main conclusion that I like presented at science fairs. But in humans, our version of ST13 is called DDX6. And so like C13, DDX6, the function of the protein is like, could be really widespread, right? If it's binding to all of these messenger RNA and choosing to lessen all of their concentrations because they've got all these non-optimal codons, it's like a really sort of, I guess, like prevalent or like important mechanism in cells because it's a way for them to like massively control a lot of protein concentrations. So, and like I said, you know, protein concentrations are really important for cellular function. So like DDX6 in humans, like mutations in DDX6 have been associated with cancers. And so I was sort of interested in this because the protein, the messenger RNA for the proteins that I looked at in my project are ones that are associated with the spindle assembly checkpoint, which will like stop cells from going through mitosis if they've got some problems. And as you know, cancer cells divide no matter what the cell's doing. They divide uncontrollably, and then they make tumors, which are really dangerous. And so if DDX6 in humans is performing this function and allowing cells to control all of these protein concentrations, then it makes sense that mutations with it are associated with cancer because it's not controlling protein concentrations. Cells don't know that they shouldn't divide, so they just keep dividing. So that was sort of the main implication of the work, I guess, and that was why it was sort of meaningful to me. But yeah.
So, are you working towards communicating your study with something that may be a journal?
Yeah, the student that I worked with during the project is writing a paper like right now about the work, and ST13 is like the paper. So yeah. And then I also, like, as part of one of my science fairs that I participated in, I had to like write a paper about it. So, you know, I've got some practice like communicating science, which I think is really valuable, and sort of at like a, I guess like a sort of communicating a high level or like complex science, which has been definitely really useful and like a skill that I'll take into college. But yeah.
Okay, so let's expand this topic a little bit. Let's talk about science communication for a while. Where do you think it fits into the structure of academia, just scientific communication?
I mean, I would say scientific communication probably fits into like every level of academia. Sorry if that's not very specific. But I think, you know, it's important to be communicating throughout the research process, even just like talking and like trying to explain what you're doing to like other members of your lab or like other labs, and thinking about, you know, what your research means, you know, where you should go from the point that you're at. I think a lot of those things can be sort of thought about while telling someone about it. You know, you can answer their questions and think, oh, you know, that's a really interesting point. So I think communicating your science is something that has to happen throughout the research process. But scientific communication is probably like, I guess, most well known, for after the research process, you've finished, you're trying to write a paper or give a presentation, and now you've got to explain your science, and you're feeling overwhelmed because you don't know how to explain your science. Um, but yeah, I think at that step it's most crucial, because you want people to know what you did and to be able to build on it, because I think that's the whole point of science, standing on the shoulders of giants and all that. But yeah, so I think, and I think communication is often like really overlooked. Scientists think, I am a science person, I am an engineer, or, I am a biologist, and I don't need to talk to anyone, I just do my science or my engineering all day. But that can't be true, because ultimately you're going to have to tell people about what you did, and you're going to want to collaborate with people maybe who are doing different things than you to reach a more meaningful conclusion. So I think we have to really emphasize scientific communication, which I think like the science fair that I competed in did a great job with that, because we had to like write a paper and give a presentation. And I think like if we encourage young really care science, I'm sure that even like curating it for different audiences and like thinking about who is like listening to them, I think that's a really valuable skill that people will need if they want to do good research and good science later in life.
Okay, so you mentioned it was overwhelming. In your personal experience, what would you say has been the proportion of effort you've put into the science and the communication? So is it like a 50-50 or a 70-30, the science and the communication?
I'm I'm not really sure how to answer that question. It feels like probably maybe like a 70-30 throughout the research process of like, trying to explain it to my friends and like talk it with my parents or talk about it with members of the lab. And that's always a good experience, like what I talked about. But I mean, the majority of my time was definitely spent like doing the research. But then in the last, month, in the last 2 months, I'd say it's like 50-50, because by then like you're trying to get all your figures together and thinking about how you're going to present. And then in the last like month, I'd say it's like 100% communication, because you're done with your project, and I just have to think like, okay, throughout this process I've sort of explained it to people, like I know what story I want to tell with my figures. Now I need to like really think, is this a story that people are going to want to hear? And if not, how can I make it a story that people want to hear? And I think, yeah, so like towards the end, I'd say it builds up the percentage that is communication.
Yeah, definitely. I like the word you used. Academic writing is definitely a form of storytelling.
Yeah. Yeah, I've been kind of interested in that. I want to take a course at some point at Stanford that's about storytelling, and try and think, how do we tell stories with science? Because I think sometimes it's like, I think especially when we're trying to communicate with like, high school students or like even younger than that, it's important to present science as like something that's fun, or like research is this, exploration, and you can discover new stories and, tell new things. But even in like scientific papers, people refer to like the progression of figures and text as like that paper's story, or say like, oh, really this research paper had 2 stories and I was disappointed that they didn't make it 2 papers. And I think the fact that we use that word really says a lot about like how much of scientific communication is just like trying to tell a story about your work.
That's
Definitely.
really well put. Especially I like the part where you talked about the different stages of communication. Yeah, I mean, it's definitely a progression and not just like stagnated at one point. So coming back to your work in general, you have also looked into another protein that plays a role in cell size control. So maybe you could talk about what this protein do.
Yeah, so my senior year in the lab, I like worked on a different project. So I switched from like messenger RNA to how do cells control their size. And it's actually a really interesting question in biology. You'd think like, oh, we've been studying biology for so long. We must know how cells control how big they are, but we actually really don't. But we know that it's really important. So like if cells are too big, I'm sure you know, like the surface area to volume ratio gets really off, so they can't transport anything, they can't take up enough nutrients, stuff like that. And if cells are too small, this has been actually associated with a really fast division cycle, which is typical of types of cancer. So there's like small cell carcinomas that are entirely defined by the fact that they're small cells. And a lack of control over cell size has also been associated with other diseases. So brain disease, cardiac disease, and musculoskeletal disease are characterized by cells that are like very different sizes or that just aren't maintaining control. So I researched this protein in fission yeast that had been found to have its concentration change over the course of the cell cycle. And this is really rare for proteins. Most of them like maintain a constant concentration. You know, they increase in abundance as the cell increases in size, but the concentration of the protein stays constant. But this protein, it increases in concentration as the cell grows. So what I wanted to know was like, you know, okay, this protein is doing something sort of weird. Is this protein potentially increasing with cell size rather than just like over time? And is this increase, you know, if it is increasing with cell size, then this protein is probably like a sensor of cell size. You know, it reaches the threshold concentration and the cell thinks, oh, this means I'm big enough. Now I'll divide. And so that could be a really easy way for a cell to like see how large it is. So my research was sort of like combination computational and wet lab. So I made like a computational model of like if this protein is accumulating with cell size, what would that look like? And like sort of some characteristics of the protein that would have to be associated with that. And then versus if it's not acting as this like really cool cell size sensor and it's just accumulating over time, what characteristics would it have there? And then I investigated these characteristics in the lab, and what I found was that it's probably accumulating with cell size, which is really exciting because this makes it like a great candidate for something that helps cells know when they divide or when they need to divide. So that was my research this year, and it was pretty exciting. And then this summer, I'm sort of continuing that work computationally.
Right. So I'm just curious about what you mentioned about the small cell carcinomas, if I got that right. Was that the focus of your work, or was it about like cell size in general?
No, I wasn't specifically focusing on small cell carcinoma. It was just sort of like cells that are the wrong size or that like lose control over cell size can have a lot of different problems, and small cell carcinoma is just one of them, but all of the other diseases that I mentioned are other ones. So like, I mean, again, you know, I was doing basic science research. Like I wasn't, you know, focused on, you know, can we disrupt this protein somehow, or like, can we control it and then prevent small cell carcinomas from dividing so quickly, or something like that. Like that wasn't what I was doing, but that would be a really interesting project to try in the future. But yeah, I was also working with fission yeast and not, I mean, like I wasn't working with like human cancer cells. It was just all in fission yeast. So yeah.
Okay, and you mentioned that it was a combination of wet lab, wet bench, and I think it's called in silico, if I'm not wrong, computational stuff. So yeah, have you been able to continue any of the wet bench research because of the pandemic, or like has it hit, or was that work done entirely?
Yeah, so I had finished the like wet lab procedures that I wanted to do, like in order to, you know, sort of present my project. I'd finished those during the school year, but I had planned to work for the lab in person and do some other procedures with the student that had been mentoring me, and do that over the summer. But then obviously the pandemic hit. Virginia Tech like put regulations in place so like I couldn't go into the lab, which totally makes sense. You know, the lab itself was like shut down completely for quite some time, but now like I'm still not allowed back, but it's okay. So I sort of pivoted and I'm doing something sort of different now. So I'm doing computational research, which I think I mentioned as something that I really enjoyed. But what I'm doing now is looking at ribosome profiling. So ribosomes are like the things that make proteins on messenger RNA. And one really interesting piece of information that you can use to learn about how proteins are made is to see where ribosomes are on messenger RNA, because then you can learn, you know, where these ribosomes may be stopping for some reason, or like this protein has more ribosomes on its messenger RNA than something else, and like what does that mean? And it's obviously like a really big data set because you like profile every ribosome in a bunch of cells. So a lot of these data sets are out there, online, and you can just get them for free and analyze them yourself. So I got a data set for fission-yeast ribosome profiling from one paper, and I've been analyzing that in sort of like connection with the research that I did my senior year. So sort of looking at information about that protein and like how it's being translated. So that's been my work this summer, and it's still been really rewarding, I guess, to like during a pandemic, be able to still do science. And I've really enjoyed like seeing what computational science looks like, I guess, because I mean, part of it my senior year, but it was always like, I will make this computational model with the intent of going into the wet lab. But now, I don't have the intent of doing that. So I'm just doing computational research fully, which has been, it's been good, despite the pandemic, to still do science.
Yeah, it's important to be adaptive. I think, yeah, science does not stop.
Definitely.
Life definitely does not stop.
I think a lot of labs would also open up virtual positions at this point. I think we were talking about this before the interview, but yeah, I think that's an important thing to note here. So yeah, so on that note, like when we were just talking about labs and approaching labs in general, did you have a mentor throughout all of these investigations, like somebody who helped you out? You can address this for like all of the projects you've done.
Yeah. So my lab is like run by a professor at Virginia Tech. And then students in the lab who were like above me sort of took me under their wing and like helped me learn about things in the lab, which was really great. So like I said, my junior year, I was mentored by an undergraduate student. And then my senior year, was mentored / I'm sort of being mentored over the summer by a PhD student. And she's doing her PhD actually about cell size, but it was funny because I had decided that I wanted to do a project about cell size control. And then she like came into the lab and had made like sort of switched her PhD to be about cell size control. So it really worked out in terms of like who my mentor ended up being. And so now I'm still sort of working with her because my ribosome profiling work is sort of in relation to the cell size control work. But yeah, I definitely like needed a mentor. The science fair rules for the ones that I participated in are like you can't just be in the lab by yourself at like 9:00 at night, putting cells onto a slide or something. So like you have to be supervised, which makes sense. And then I also like needed help, learning about how to do all the procedures and, using the microscope and stuff. But I do want to stress, like, research doesn't have to be something that you like go into a lab, or like you don't have to have access to a university or a lab like that in order to do research. I mean, maybe you can't do the kind that needs like a PCR machine, but you can definitely just like look in your own community and, see a problem and think, how could I solve this problem or address this with science and engineering? And, if you have internet access, like my research this summer was just done totally, like I read some papers about ribosome profiling, found this data set, and started working on this protein that is interesting. So that was like entirely through the internet, and like you can contact a professor remotely and say, I'm interested in your work, due to coronavirus, your lab has probably had to go online. Is there any way that I could take part in your research? So, if you have internet access, you can do meaningful science.
Yep, definitely. So, do you feel like you've ever had to carve out opportunities for yourself through cold emailing, or I mean, have you had to spend a lot of effort doing that compared to your research work, or has it been something that in front of your research was just like, I suppose, a pause before you move on?
Wait, I'm sorry, like coding? Did you say for coding?
I'll just like reframe the question. We can cut it out. I mean, do you feel like you've had to spend a lot of effort cold emailing people and just carving out opportunities for yourself compared to the effort that you spend on research?
Yeah, I would say no. I was really lucky in that, my first round of emails, like everyone said no, but led to a spot. at a lab. And then I decided to stay at my lab. I think if I decided to switch labs, it probably would have been a bit easier to get into a different lab, because I would have had this PI, professor, principal investigator, who like knew me and, you know, could recommend me and say like, this isn't just a random high school student, you know, she's worked in my lab. But I think I definitely got really lucky. And I decided to stay in my lab because I really liked the community. Everyone in my lab is really supportive, and really willing to like take some time out of their day to like answer a question that you have, or like help you out with some code that's not working or something. And I also knew a lot more about like fission yeast and like the procedures that were available to me. So I was able to sort of like do, you know, I knew a lot more about like what I could do, and, you know, could design a question much more efficiently. So that's sort of why I decided not to switch. But I think had I switched, you know, yeah, some of the time between my junior and senior year, like that summer would have been spent in sort of emailing / trying to get a new spot. But yeah, I was lucky in that not a lot of my time was spent doing that.
Okay. So, touching back upon your project, you said that the intent of the computational work that you're doing is no longer to do wet bench work. So, can you talk about what plans you have for the future to take this work forward?
Yeah, so I've like made some findings with the data set that I've got, and I'm right now trying to get another data set to try and confirm that. So that's sort of the major thing that I'm working on right now. Ribosome profiling is like a pretty new procedure. And there are a lot of studies like ribosome profiling in mice or ribosome profiling in budding yeast, but there isn't a whole lot with fission yeast. So I'm just right now trying to get that other data set to sort of confirm what I found. And then some other next steps, I have some more research questions that aren't associated with the protein I've been working with that I also want to look in this ribosome profiling data set to answer, because like I said, it's a pretty new procedure. So there are a lot of like still open areas and like questions to answer that are associated with it, which I think is really exciting. So that would be along the lines of like, similar to what I did junior year, like there are some findings with budding yeast, and I could try and confirm that with fission yeast to see if it's a widespread mechanism. But yeah, those are sort of my future plans, I guess.
Well, good luck.
Yeah.
Thank you. Thanks.
With this, we are at the end of part 1 of a 2-part series where we talk to Claire Morton. You can find the second part of this episode on all leading podcast platforms such as Apple Podcasts, Spotify, and Google Podcasts under the name of a STEM Research podcast. Link is added in the description. Thank you, Claire, for joining us today, and our audience for tuning in. We hope to see you in our future episodes.